next presentation. And the annual review lecture is the prize lecture that the Society holds in the highest regard. This is our main award lecture for the annual meeting, which we're at today. And the annual review lecture is given to an eminent physiologist, and today's recipient is no exception to that. This year's awardee is Professor David Attwell, who's the Dodrell Professor of Physiology at University College London. David Attwell started his academic work as an undergraduate at Oxford, where he read physics and physiological sciences, and undertook a DPhil with the biophysicist Julian Jack. For his DPhil, he won the Gotch Memorial Award for the best thesis, and then subsequently went to Berkeley to do a postdoc. He then came back briefly to Oxford, and in 1981 was awarded a university lectureship at UCL, and since has gone through the ranks of that institution. And in 1995 was made a Dodrell Professor. En route to this major chair, David Attwell has achieved much distinction in the area of neurophysiology. He is well renowned for his work on neuronal glial interactions and glutamate transport systems. And this work has underpinned several major honors that he has received. He won the Sharpie-Schaefer Medal of the Society in 1986, and in 2000 was elected a Fellow of the Academy of Medical Sciences. The following year, he was elected as a Fellow of the Royal Society, and then became a member of the Academy Europea. He's also a member of the Norwegian Academy of Science and Letters, and he is one of the Web of Science's most highly cited scientists. Today, we're going to hear some of his most recent work, in particular, the role that pericytes play in blood flow control in the cerebral circulation. So please join with me in welcoming this year's Annual Review Prize lecturer, Professor David Attwell. David, over to you.
Thank you for that kind introduction, David. Today, I want to try to convince the audience that there's been a kind of revolution in our understanding of how blood flow is controlled at the tissue level. In particular, the fact that these tiny cells, pericytes located on capillaries, can regulate blood flow not only in physiology, but especially in pathology. I'm going to give you an overview, first of all, of what I'm going to talk about. I was asked to briefly mention my career path. I'll then get into the meat of the lecture on the regulation of energy supply in the brain. I'll show you that pericytes can constrict and dilate capillaries, and I'll show you that the signaling for regulation of capillary diameter differs from that for regulation of arterial diameter. Then I'll turn to the dark side and look at the role of pericytes in ischemic pathology of the brain, first of all, and then of the heart. I'll show you that pericytes play a key role early in Alzheimer's disease, and I'll suggest that they have a role in COVID-19 as well. And throughout all of this, I'll bear in mind some therapeutic approaches. So let's start out with career path. I started out, as David just mentioned, doing physics in Oxford, where I lived at the bottom of this large tower here. The bells were rather noisy. And I was all ready to do a PhD in plasma astrophysics, studying things like this, when my medical colleagues persuaded me that biology was more interesting. So I ended up doing a PhD with Julian Jack, who very kindly took in a refugee from physics. And I mostly worked on the properties of neurons, but also on the heart, in part, in Dennis Noble's lab. Things were different then. Hair was longer and browner. But eventually, I escaped from Oxford and went to the Bay Area to do a postdoc in Berkeley, where I worked on information processing in the photoreceptor layer of the tiger salamander. Coming back, I rapidly got, things were easier back then, I rapidly got a lectureship at UCL, where I've been for the last 40 years, studying many things about the nervous system, but especially neuron-glial interactions. That's how neurons interact with cells like these microglia, oligodendrocytes, and astrocytes. So with that background, I want to now look at energy supply regulation in the brain, because this has been a major focus of what I worked on for the last 20 years. Homer's brain is 2% of his body's mass, but it uses 20% of his resting energy production. And that's why even Homer has a very dense network of blood vessels like this, bringing in oxygen and glucose to power the neurons. For those of you who are more tissue slice focused, this is a hippocampal slice. These little bobbly things are the neurons, but you may be unaware that to make all of this work, this little piece of brain needs this dense network of blood vessels to bring in the energy. And even then, the oxygen and glucose supply coming in through these vessels can't keep all of these cells working at their maximum rate all the time. So dilations have to occur locally around active neurons in order to power them. So how does this happen? In the cortex, blood comes down from the surface of the brain through penetrating arterioles, which are ringed by smooth muscle. And conventionally, it's assumed that changes of the tone of these smooth muscle cells alter the diameter of the arterioles, thus affecting the amount of blood which goes into the capillaries, where nutrient exchange occurs with the neurons. But it's really not the whole story, because back in the 1870s, these guys, Karl Ebert in Germany and Charles Rouget in France, discovered that there were these cells present on capillaries which were potentially contractile. And I'll show you they are contractile. I want to digress for a second on the rise of studies on pericytes. As I mentioned, they were discovered in the 1870s. They were only named pericytes 50 years later, but it's only recently that there's been enormous interest in their properties. Now, more than one paper mentioning pericytes in the abstract is published every day, but they remain seriously understudied. They're present at roughly 50 to 150 micron intervals along capillaries, so they're a major cell population in the brain. But if you go to PubMed, you'll find that only one brain paper in 1,000 mentions pericytes. And it's even worse in the heart. Pericytes are present on all capillaries in the body. In the heart, they're actually the second most numerous cell type after endothelial cells. Myocytes come a poor third. But in PubMed, only one cardiac paper in 5,000 is about pericytes. Parasites probably contribute to all diseases involving ischemia, like stroke, heart attack, spinal cord injury, as well as we'll see to Alzheimer's and COVID, as well as diabetes and cancer. But they're largely ignored both by scientists and big pharma. This implies we're missing therapeutic opportunities. So I'm a kind of missionary for parasites, pushing their importance. I want to start by showing you that parasites can constrict and dilate capillaries. It's an almost unknown fact now that in 1920, a Nobel Prize was given to August Crowe for the discovery of contractile cells on capillaries. Here he is sitting in his armchair, and this is his diagram of skeletal muscle and his estimate of the variable proportion of capillaries which were open and closed at some particular energy demanding level of the muscle. His work has been almost completely forgotten from the literature, apart from certain historically minded Danes, of course. More recently, there has been a lot of work on retinal parasites by Don Puro's group in the first 10 years of this century. But still, very few people, some people still claim that parasites cannot regulate capillary diameter and they're not contractile. So I want to start off by showing you a movie to prove that they do. This is a capillary in the retina. This patch pipette is located on top of a parasite here. There's another parasite here and one here. They're a bit hard to see in the image. When we pass depolarizing current through the pipette, and this was an experiment done by Claire Howard, then a PhD student in my lab, you'll see that the parasite completely squeezes the capillary till it's totally occluded, showing their power to control blood flow. If I play the movie again, you'll see that after this central one has constricted, the parasites on each side constrict as well. And that's because an electrical signal goes along the endothelial cells from one parasite to another. And you can see this in this plot here of the diameter at various locations. The central parasite repeatedly constricts each time it's stimulated, and so do the ones on each side with a certain lag. But in between, there's no constriction of the capillary. So it's only the pericytes which are the contractile cells on capillaries. Now, it turns out that neurotransmitters also regulate capillary diameter via pericytes, and I want to show you this for human tissue. We get human tissue as a byproduct of neurosurgical operations. So if you want to access a glioma to remove it, then you typically have to remove some normal cortical tissue above it. We get that tissue and cycle it around to the lab, and then we can use it for histology or live human brain slices. Here you can see a capillary in one of these slices with the pericytes located on it. They're outlined by this label isolectin B4, which labels the basement membrane around them. If you look at them when they're alive, you can see a pericyte here sitting on a capillary. These are red blood cells inside the capillary. And if we apply, for example, noradrenaline, it constricts the capillary near the soma. Here's the diameter going down. If we apply glutamate to increase neuronal activity, they dilate, and eventually this washes out again. So pericytes constrict capillaries near their somata, near the somata of the pericytes. Why is that? Well, it's to do with the geometry of the cells. It's only circumferential processes that can squeeze the capillary. And if you look at transgenically labeled mice in which pericytes are red, you can see that there are circumferential processes mostly near the soma. They also have longitudinal processes going along them. And in these mice, we counted the percentage of circumferential processes at different distances from the soma, and nearly 80% of those circumferential processes are within 10 microns of the soma. Accordingly, if you apply a constricting agent like this thromboxane A2 analog, U46619, you get a big constriction at the soma, but a more mild constriction further away. And you can plot this in a way which will become useful later by plotting the diameter of the capillary as a function of distance from the soma. And in the control conditions, when they're relaxed, the diameter is actually larger near the soma because the soma interacts with the endothelial cells to make the endothelial tube grow, whereas after you apply the constrictor, there's a specific constriction at the soma without much effect far from the soma. And I want you to try to remember this diagram because it's going to recur in the context of Alzheimer's disease. So the ability of pericytes to constrict capillaries really matters because it turns out in the brain, at least, it's the capillary bed which offers the largest hemodynamic resistance to the blood supply. So anything which can control the diameter of the capillary bed has the power to adjust blood flow. We wanted to look at this in vivo, however, not just in brain slices, because of course brain slices don't have blood flow or blood pressure. And to do this, we use 2-photon imaging in vivo through a hole above the somatosensory cortex. And we can then evoke neuronal activity by stimulating the whisker pad of the mouse this was done in. And we can then monitor the diameter of the arterioles and the capillaries at the same time. This top left diagram over here shows a penetrating arteriole going down into the board. The green stuff is phytidextran in the blood. And here's the first order capillary coming off it, splitting into 2 second order capillaries. And this red bit here is the pericyte wrapped around it. Now, if I play the movie, I want you to look here, first of all, at the capillary, because when we stimulate the whisker pad, the first thing that happens is the capillary dilates, and only later the arteriole dilates. The time course is at the bottom here. I'll play it again. The capillary dilates, and then the arteriole dilates. This is a carefully chosen example. In the time difference between them is about 3 seconds rather than the mean value of 1 second. But it's important because it shows the capillary relaxes and at a time where the arteriole has not relaxed at all. So it's not just that the capillary is being pushed open by a local increase in blood pressure, which occurs when the arteriole dilates. The arteriole hasn't dilated at the time when the capillary is almost completely dilated. And if we look at the size of the dilation, so it's faster in capillaries, what about the magnitude? Arterioles dilate on average by about 6. If you look at the capillary branches nearest to the arteriole, they dilate a bit more, about 8%. And as you go further into the capillary bed, the dilation gets somewhat less. And that's partly because they have less circumferential processes and partly because they have less contractile proteins in them. But on average, they dilate about 7%, slightly more than the arterioles. So low branch order capillaries dilate faster and more than arterioles, and that means they probably play a key role in increasing blood flow when neurons are active. This means that they contribute to the bold fMRI response, which reflects that increase of blood flow. Okay, so pericytes can constrict and dilate capillaries, but do they do it in the same way as occurs for arterioles? I don't have time to show you all the data, but the answer is no. There are different signaling pathways controlling these two types of vessel, as is shown in these papers down here. So when glutamate is released from active neurons, one way in which it dilates vessels is it acts on NMDA receptors on interneurons. The calcium influx activates nitric oxide synthase, and that releases nitric oxide, which diffuses to vascular smooth muscle cells, raising cyclic GNP concentration in the classical manner and relaxing the vessel wall so the arteriole gets bigger. The other pathway is that glutamate activates a release of ATP from postsynaptic neurons. This acts on P2X receptors on astrocytes, raising their calcium. That generates arachidonic acid, which is converted to prostaglandin E2, and that in turn activates a potassium channel in the pericytes, which hyperpolarizes the cell, closing voltage-gated calcium channels and relaxing the pericyte. So there are these two separate pathways contributing to the increase of blood flow which occurs. And this is all done in the context of a background constriction produced by locus coeruleus axons releasing noradrenaline. So that's the kind of physiological background to the main part of my talk, which is going to focus on various pathologies of the brain and also a little bit the heart. I want to start out by talking about the role of pericytes in ischemic pathology of the brain. Back in the 60s, Adelbert Ames did a classic experiment involving ink. That's the kind of stuff you would write letters with back then. So what he found was if he injected India ink into the blood of, I think it was a rabbit, it would go through all the blood vessels in the rabbit, and you could see them all nicely as is shown here. However, if he initially did middle cerebral artery occlusion in the rabbit for an hour, and then opened up the artery again to let the blood flow back through the artery, when he injected the ink then, he found it only went into the larger vessels. The smaller capillaries were often not perfused by the ink. And if you look at the whole brain here, he found that there were these white areas where there was very little perfusion occurring after a period of ischemia. So following ischemia, blood flow returns to arteries, but some capillaries fail to fill. Now, why does this happen? We looked at this by imaging, again, capillaries in the retina and imaging pericytes like this one here, and we measured the diameter of the capillary at the pericytes. And when we superfused solution which mimics ischemia by blocking ATP production from glycolysis and mitochondria, we found that the diameter of the capillaries decreased. Now, this is not surprising. If you think of this as a pericyte with its arm around a capillary, normally lots of iron pumping is done powered by ATP. When you cut off the energy supply, you lose ATP, pumping stops, and that leads to a rise of calcium concentration, which activates the myofilaments in the pericyte and makes it squeeze the capillary. This also happens in vivo after middle cerebral artery occlusion. Here we've applied MCAO to a mouse in which there's physialbumin in the blood in order to see the perfused vessels. The vessels have been labeled with this far red conjugated isolectin B4, which is painted mauve here. And the pericytes are just visible as DS red fluorescent pericytes. And what you can see is that after the MCAO for 90 minutes and then reperfusion, so there is blood flow back through the middle cerebral artery, lots of the vessels have blood come back into them, but there are vessels present which have no blood in them. We wanted to know why this is the case. And to do this, we put lots of little lines. You can need a graduate student to do this. You put lots of little lines across the vessels and you measure the diameter at all those places. And what you find is that both in the core of the lesion and in the penumbra and the cortex, there's a specific decrease of diameter, which is larger near pericytes than away from pericytes. And this is what's restricting blood flow. And in some cases, it cuts off the blood flow completely, which is why we have those vessels which are no longer perfused. Now, other people have monitored the blood flow through this kind of process, and they find that when the middle cerebral artery is occluded, of course, the blood flow goes to zero almost. And on reperfusion of the artery, there's a period of reactive hyperemia, but then the blood flow settles down to a value which is only about 60% of what it was to start with. So there's a long-lasting fall of cerebral blood flow. Why is this so long-lasting? Why isn't there a dilation of pericytes again when the oxygen and glucose supply is restored? And we think we found the answer for this by imaging cortical slices. If you image cortical slices for a long time in control solution, then you find that there's no change in their diameter. Whereas if you do it applying ischemia solution, this is chemical ischemia again, so a very severe insult to deplete the ATP, you find that the diameter near pericytes goes down quickly, as you can see by these shrinking red lines, as are shown here. And if we then apply propidium iodide after about an hour of ischemia, propidium is normally excluded from cells because it's got 3 positive charges, so it won't cross the membrane. But if the membrane ruptures or becomes compromised, it can then get into the cell and label the DNA and fluoresce. And we found that after about an hour of severe ischemia, lots of the pericytes died. So what they've done is they've constricted, but they've then died in rigor. And they die in rigor because there's no ATP to let them relax. If you remember your muscle 101, muscles need ATP to relax, not to contract. And so the pericytes become stiff like a corpse, and they can't relax, and the capillary will stay squeezed by the pericyte. Here's the time course of this. This is the constriction in black. This occurs with a half time of about 15 minutes. And the death of pericytes occurs with a half time of about 45 minutes. And after an hour, they're almost all dead. So what we think is happening, this occurs in the white matter as well as the gray matter, as I've just shown you. What we know is happening actually is this rise of calcium, which I mentioned before, triggers the death of pericytes. So if we remove the calcium from outside the cell, then you significantly reduce the amount of pericyte death occurring. So they die in rigor, they produce a long-lasting constriction. This is what's conventionally called the no reflow phenomenon. And it's lack of ATP, which prevents the pericytes relaxing. So because of this long-lasting decrease of blood flow, this may reflect the death of pericytes, and that makes pericytes a therapeutic target in stroke. And to start to address how one might stop this happening, if you're trying to treat stroke patients who've been brought into a rapid treatment stroke center, for example, we've collaborated with Alistair Buchan's group in Oxford to look at the effect of reducing the calcium influx into pericytes, either during ischemia or more useful clinically after the ischemia. So if you have nimodipine present to block the pericytes L-type calcium channels during ischemia, you find that slows the constriction which occurs predictably because the calcium rises more slowly. If you use oxygen glucose deprivation instead of chemical ischemia, so you can remove the insult after an hour, you find that although during an hour's OGD, there are about 50% of the pericytes die, another 20% die during an hour of reoxygenation. If you do all of this with nimodipine present, you significantly reduce the pericyte death occurring. Now, what effect might these contribute in vivo? We've given nimodipine on reperfusion after MCAO to see what happens. There's no point in giving it before the MCAO, really, because clinically, you're only going to be able to give drugs when the patient knows they've had a stroke. So if you measure the blood flow, then normally during the MCAO, blood flow is greatly reduced. And when you open up the artery again, blood flow increases, but never to the control level. Whereas if you give nimodipine at the moment of reperfusion, then you find that blood flow does come back, and in fact it overshoots the original value. So this looks promising. And if you assess behavior, assess behaviorally how well the rats perform after this has been done to them, this is an assessment a week later, I think, you find that their neuroscore is much lower, significantly lower for the nimodipine than for the vehicle-treated rats. So targeting parasites can be a useful therapy. A similar ischemic pathology occurs in the heart, and I want to turn to that now. I showed you these pictures of no reflow in the brain after ischemia, but exactly the same thing happens when you have a heart attack, or can happen. So this is something like ink going through all the vasculature organization in the heart is very different to in the brain. There are many, many parallel capillaries, each with myocytes in between them. Ink goes through these vessels normally in the normal heart, but after you've occluded the left anterior descending coronary artery for an hour and then let it open again, you find that the ink doesn't go through a lot of the capillaries. And this is a major clinical predictor of bad outcome. So could parasites be involved? If we look at a picture of the left ventricle of the heart, here green is labeling the capillaries and red is labeling the parasites. You can see much of the picture is yellow, showing that parasites are all over the vessels. In fact, they're the second most common cell type in the heart. And here's one which is more spatially isolated so that you can see it puts these circumferential processes around the vessel and might squeeze them. So could they be involved in this no reflow phenomenon like they are in the brain? To look at this, we put green albumin into the blood and then fixed hearts and just measured the amount of green stuff at various positions around the left ventricle. This is a section through the heart. This is the right ventricle, this is the left ventricle. If you go round clockwise from the middle of the septum, you get a density of green stuff which follows this rather wobbly curve for the control situation, which I've approximated by this straight line. If you then do it after ischemia, so you occlude the left anterior descending coronary artery for an hour, and then open it up again for, I think, 20 minutes, you find there's a long-lasting decrease in the amount of phytidextran or phytialbumin on this side of the heart. That's shown as this black line here. Interestingly, this fall of perfusion is ameliorated if you apply adenosine at the moment of reperfusion. Adenosine is an agent known to relax parasites, which it does by raising cyclic AMP. If you focus in on these images, as I'll show you in a minute, there's hardly any blocked in control, any capillaries blocked in control conditions, but after ischemia, about 40% of the capillaries are blocked, thus accounting for the no reflow. And this again is reduced to 30% if you add adenosine at the moment of reperfusion. Let's look at those capillaries. Here you can see green blood flowing all the way through the image in some capillaries, but for other capillaries, the green blood stops at an apparent site of block. And looking at the parasite label, you can see it's near the pericyte-soma, and it looks as though the parasite processes have squeezed the vessel to stop the green stuff going through. So the concentration of green stuff, green phytidextran, falls dramatically across that boundary. And if you measure the distance from the site of block to the pericyte-soma, which is where most of the circumferential processes are, you find the blocks are mostly within about 5 microns, or within 10 microns of the soma. The half point here is 5 microns, whereas the red line shows what you would expect if the blocks were randomly positioned. So this very significant difference tells you that the blocks are associated with parasites. And if you measure the diameter of the capillaries at the pericyte-somata, they go down from about 5.5 to 3.5 microns after ischemia and reperfusion, and adenosine increases that diameter again. So we think that in the heart, like in the brain, parasite constriction produces no reflow, and it not only directly reduces blood flow, it may produce sites where leukocytes or red blood cells get stuck, increasing the amount of block. So we think they're also a therapeutic target in the heart. I want to turn now to the role of parasites in Alzheimer's disease. You're familiar, I'm sure, with the fact that amyloid beta accumulates in Alzheimer's disease as you go from being a healthy control through mild cognitive impairment to late onset AD. And tau gets phosphorylated, but it's less well known that the first thing which occurs is there's a decrease of cerebral blood flow, shown as this brown curve here. And in fact, Alzheimer's is associated with roughly a 45% reduction of cerebral blood flow in affected areas. Does this fall of blood flow matter? Well, it certainly does. If you have normal blood flow, you have normal function. When you have a 20% decrease of blood flow, you suffer a loss of attention, possibly due to adenosine release suppressing synaptic transmission. But when you get down to the range occurring in Alzheimer's disease, you start to inhibit protein synthesis. That's obviously bad news. You have damage to myelinated axons, spatial memory deficits, inflammation kicks in, and then you get synapse loss and neuronal damage. This is all well before you reach the very low levels of blood flow which occur when you have a stroke, which causes glutamate release, the anoxic depolarization, and tissue death. So we wanted to look at this in humans, and we took advantage of the fact that at Queen Square, if you're dementing for no known reason, they will sometimes take a live biopsy from your right prefrontal cortex, an area neurologists deem to be apparently of very little use, and they then label that tissue to see whether you're suffering from Alzheimer's disease or some other disease that might be treatable. So they label for A-beta and phosphorylated tau. Half the patients have no A-beta deposition and are not getting AD, and the other half do have it. We can then access that archived tissue and label for pericytes with an antibody to PDGFR-beta and look at the diameter of the capillaries. And when we do that, after measuring about 9,000 capillary diameters, again, a graduate student is a good investment here, you find there's about an 8% decrease in the mean capillary diameter, which may not sound much, but if that was true everywhere, it would decrease blood flow by 30% because of Poiseuille's law. Flow goes as the fourth power of the diameter. But to pin what's happening on pericytes, we look at diameter as a function of distance from the pericytes, as I mentioned before. And we found, just like I showed you earlier, that in patients not depositing A-beta, the diameter is larger at the soma, and in ones who are depositing A-beta, there's a big 30% constriction at the soma. And this difference in profile predicts a 50% decrease in cerebral blood flow. I just want to remind you, this is very similar to what I showed you earlier in brain slices applying a constricting agent. So the diameter is specifically reduced at the soma because that's where the circumferential processes are. So we think the diameter is larger at the soma here because the soma induces growth of the endothelial tube. Alzheimer's disease results in constriction at the soma. We can look at this as a function of the amount of A-beta being deposited. And if you look at this, as A-beta starts to be deposited, the extrapolated diameter at the soma very rapidly reduces here. So this tells you that this is a reduction which will occur early in AD and therefore may be involved in kicking it off. We sent this to a nice journal and they said that's all very well doing it in humans with AD, but wouldn't you like to do it in transgenic mice? So we had to do it in transgenic mice, and we used this model here. And we found again there was a decrease in the diameter. In general, it was larger at the soma. And again, we got this now classic picture where there was a specific constriction at the soma. These constrictions may trap neutrophils, adding to the reduction of blood flow, as seen by Chris Schaefer's group. And we saw no such constriction in AD patients, in AD mice, when we looked in the cerebellum where A-beta is not deposited. Interestingly, at the endogenous level of A-beta occurring in these mice, arterioles and venules are not constricted. So it's definitely the constriction of the capillaries which is reducing brain blood flow. What's the mechanism? If we go back to our live human brain slices, make A-beta oligomers with roughly 4 times the monomeric weight and apply them to these slices. This is obviously a crazy experiment to do because we're trying to mimic something which takes decades in about an hour. But if we apply A-beta, we find there's a slow constriction of the capillaries. And we believe this is relevant because of what I've shown you in the actual patients getting AD. And if you look at the EC50 for this effect, we find that it's about 5 nanomolar, which is similar to the level of soluble A-beta in AD brains. The mechanism, which I don't have time to show you, is that A-beta generates reactive oxygen species, which releases endothelin and that acts on endothelin receptors on the pericytes, which dumps calcium from internal stores, activating the myofilaments to constrict the capillary. We can try to block that by blocking ROS production or blocking endothelin receptors, but blocking endothelin receptors is known not to be a good way to reverse what they're doing once they've already been activated. So we also used another approach, applying C-type natriuretic peptide, which activates a guanylate cyclase receptor, which makes cyclic GMP, and through PKG that suppresses calcium release from stores and may block the constriction. It also, by a Viagra kind of mechanism, raises cyclic AMP, which activates myosin light chain phosphatase, which will remove the phosphate that's activating constriction. So does this work? If we evoke a constriction with A beta, as occurs here, then if we apply the blockers of the activating pathway, you can stop the constriction in its tracks, but it only slowly reverses, whereas the C-type natriuretic peptide produces a rapid reversal of the constriction. So what I've shown you for this part of the talk is that A beta releases endothelin, which decreases blood flow via pericytes and decreases energy supply. This reconciles genetic evidence for the importance of A beta with the fact the earliest event in AD is vascular dysfunction. I've suggested ways of blocking this. Now, other people have shown that when energy levels fall, that upregulates BACE1, the enzyme which makes A beta. So there's an amplification going on here where more A beta will be produced as a result of this fall of energy level. But in addition, this may explain the fact that people who repeatedly suffer transient decreases of energy supply, for example, people with sleep apnea, or people whose head repeatedly gets hit, like boxers or footballers heading the ball, they tend to get A beta more than your average person, and that may be because they're kicking off this feedback loop here. The rise of A beta, which will be amplified by this, produces phosphorylated tau, and together they cause synapse and neuron loss and dementia. For me, the important question is, how much of that synapse and neuron loss is produced by these pathways, and how much is produced by the fall of energy level powering those synapses and neurons? I should have mentioned the fall of energy level promotes tau phosphorylation as well. And here's a reference for further reading. Okay, lastly, I want to talk about the possible role of parasites in COVID-19. You'll have heard of the virus. Here it is with its spike protein on the surface. The spike binds to a receptor, which turns out to be an enzyme called ACE2, angiotensin converting enzyme 2. When it binds, that leads to virus internalization and infection of the cell. As I'll show you in a second, in the brain and also the heart, the receptor ACE2 is largely expressed by parasites, with some in endothelial cells, which is why we got interested in it. But it's interesting that this virus can act all over the body. It infects the nasal pathway and the lungs, of course, but it has effects on the brain where blood flow goes down. It affects the kidney where blood flow goes down as well. It affects all sorts of organs. And the classical explanation is that this occurs perhaps via the release from the lung of pro-inflammatory cytokines. But the virus can also spread through the blood when you have a high viral load, and it can bind to ACE2 in other organs. It can cross endothelial cells via transcytosis. It gets into the brain via the olfactory tract, and brain parasites are known to get infected. So what does this do? Unfortunately, we need to revisit the renin-angiotensin system, which I had forgotten about since lecturing about it 40 years ago. This is the classical part of it. Angiotensinogen, when you need to have more body fluid or sodium, you release renin, which converts angiotensinogen into angiotensin 1, which is then converted to by ACE, a target of blood pressure pills, into angiotensin 2. This mostly acts via the AT1 receptor, causing vasoconstriction, but there's a bit of an effect on AT2 receptors, which oppose that. But this is, more recently, it's been discovered in the last 20 years that these peptides are converted eventually by ACE2 into angiotensin 1 to 7, which acts on the MAS receptor, which is a strong dilator. When the virus arrives and binds to ACE2, it's known for the related SARS virus. It doesn't occlude the binding site for angiotensin 2, but virus internalization removes ACE2 from the membrane. So we'd expect the virus to stop ACE2 working and decrease the amount of dilation via MAS. To study all of this, you can't do it with mice or rats because they don't bind the virus, at least not the normal Wuhan version of it, which we've used. It turns out there's a key lysine at position 353 in the human and hamster sequence, which is converted to a histidine in mouse and rat. So we've worked on hamsters for this work, validated by these guys showing you can infect hamster with COVID-19. If you label the brain for ACE2 and pericyte markers, you find they co-localize. And if you quantify the overlap of ACE2 with pericyte markers, you find that 80% of the ACE2 is in PDGFR beta positive cells and 80% of it is in capillaries, with a small 20% in penetrating arterioles. So capillary pericytes are where most of the action is. What does angiotensin do to brain pericytes? If you apply it, you get a constriction. That's blocked by losartan, the AT1 receptor blocker. But so that's angiotensin 2 acting on AT1 receptors here. If you block AT2 receptors or MAS receptors, you get a bigger constriction, especially when you block MAS, which tells you that these two dilatory receptors are active normally, and they're opposing the constriction produced by angiotensin. How does the virus affect this? We've used just the receptor binding domain part of the spike protein to look at this. And if you apply that on its own to brain slices, you find it produces a kind of not very convincing constriction. It is significant, but it's only slowly developing. But if you apply angiotensin on its own, it gives you this small constriction, but in the presence of the receptor binding domain, you get this massively potentiated constriction. This is admittedly a very high dose of angiotensin 2, but the effect is specific to angiotensin because it isn't seen, this potentiation is not seen if you apply a different constrictor. You get the same potentiation with a lower dose of angiotensin, and if you make a mutated version of the receptor binding domain with one amino acid changed to abolish its binding to ACE2, then you abolish this potentiation of the constriction. Overall, we see about a 12% decrease of capillary diameter in the presence of the RBD. So we think normally angiotensin 2 activates all of these receptors, but the dominant one just about is the AT1 receptor. When the virus has removed ACE2 from the membrane, there's no longer any activation of MAS. And so you get a much bigger constriction. So you ought to be able to mimic this simply by blocking ACE2, and that is exactly what happens. This black curve is with ACE2 inhibited. The red curve is applying the receptor binding domain. So this is just blocking ACE2. There's no receptor binding domain applied in that case. And secondly, you can block this constriction with losartan, and although it's not statistically significant yet, hydroxyfacidel, another clinically used drug, reduces the constriction produced by the receptor binding domain in the presence of angiotensin. So some caveats. AT1 receptor blockers might provide some therapy for some COVID-19 effects. This needs extending to in vivo data with pseudovirus. We're not sure how large an effect on blood flow will be produced, but a 12% constriction everywhere would decrease blood flow by 40%. We don't know the real angiotensin level in the brain. We don't know whether it comes from the kidney or the local brain renin-angiotensin system. I've come to the end of my talk. What I want to briefly overview is I've told you that energy supply regulation is important in the brain. I've shown you that pericytes regulated in part at the capillary level. Astrocytes regulate capillary diameter, neurons control arterioles. Pericytes play a key role in no reflow after ischemia of the brain or heart. Pericytes play a key role in initiating Alzheimer's disease, we believe, and pericytes may play a key role in COVID-19. In the future, studies of pericyte diversity are going to be key because the properties of pericytes vary along the capillary bed, and more diseases are going to be shown to have pericyte involvement, especially diabetes. Pericytes regulate blood flow in the pancreas, and they're known to contribute to diabetic effects in the retina. We're now looking in the brain and periphery, and they also contribute to glioma and epilepsy. This work couldn't have been done without an enormous team of brilliant co-workers. I'll just highlight a few. The ischemia work was led by Catherine Hall for the brain and by Fergus O'Farrell for the heart. The Alzheimer's work was led by Ross Naughtley, and the COVID work was led by Win Hiram Patarasilp. Thank you for listening, and this is a nice picture of parasites.
So,
Thanks.
David, thank you very much for an outstanding review lecture. And as a review, you covered a huge amount of territory and beautifully showed the central role that parasites play in a huge amount of physiological function. And in particular, your most recent work on the COVID story looks absolutely exciting for therapeutic opportunities. As is the tradition of the annual review lecture, we don't take questions to the award lecturer, but there is the opportunity coming up for people to meet David Attwell, and I'm sure you may have many questions for him following his lecture. If we were doing this in person, then I would be presenting you, David, with one of the commemoration statues which the Society gives its awardees. So once again, many congratulations on the award of the prize lecture, and thank you very much for an outstanding lecture.
Thank you.